Assalamu alaikum. My name is Mubashir Khalid, a student of Zoology Department at University of Sialkot. Today my topic of presentation is relationship of plasma, liver enzymes and lipid profile with type 2 diabetes mellitus in Sialkot population. First of all, I would like to elaborate on why I choose this topic. So here are some important reasons. Type 2 diabetes mellitus is one of the most common metabolic disorders. It contributes to more than 90% of all the cases of diabetes. It disturbs the normal physiology of body by deteriorating other pathways. More than 1% in every 10 adults has diabetes. It is also the risk factor for other diseases like diabetic retinopathy, diabetic nephropathy, and diabetic neuropathy. Doctors and some researchers say that if a person died due to heart attack and he also had diabetes, so one should write on his gravestone that this gentleman died due to the complications of diabetes. So this disease has a huge burden on the health sector, especially in the developing countries like Pakistan. Introduction of the study. Type 2 diabetes mellitus is one of the most prevalent forms of diabetes characterized by insulin resistance and beta cell dysfunction. This insulin resistance in the body tissues results in the intolerance of glucose, causing the hyperglycemic conditions. This hyperglycemia affects different organs, especially the liver and its enzymes. Liver enzymes are very important and they control the metabolic conditions of the body and play a very major role in glucose transport, and their dysfunction results in high condition in blood glucose level in the blood, which may worsen the glycemic conditions further. Aims and objectives. To investigate the concentration of AST, ALT, and ALP in type 2 diabetes patients, Sialkot. To determine that insulin resistance is responsible for lipid abnormalities. Let's discuss first how normally the transportation of glucose occurs. Then we will talk about the changes in type 2 diabetes mellitus. So when we eat food, the glucose from the GIT is absorbed into the blood and the blood glucose level rises. This glucose will move from blood into the interstitial fluid and then from interstitial fluid into the beta cell. Beta cells of pancreas have potassium channels which are mostly leaky, and potassium ions move outside of the cell through these channels, but due to the breakdown of glucose, the formation of ATP will phosphorylate these channels and these channels will be closed. After their closure, the potassium ions will not migrate outside the cell and the inner side of the cell will become more positive, which will open the voltage gated calcium channels and the calcium ions will move through these channels. When calcium ions moves inside the cell, they will stimulate the release of insulin. Most of the tissues in the body have insulin receptors, and this insulin will bind with these receptors, and there will be a beginning of an intracellular cascade, which results in the deposition of glucose transporters on the plasma membrane. When these glucose transported are deposited on the plasma membrane, then glucose will move from blood into the body tissues. But in type 2 diabetes mellitus, which may be due to metabolic syndrome, first degree relative, or ethnicity, The cells become resistant to insulin, and there will be no glucose transporters on the plasma membranes, and the glucose transportation from the interstitial fluid from blood or interstitial fluid into the body tissues will be inhibited, which results in hyperglycemic conditions. The high level of glucose in the blood triggers the beta cells to release more insulin, along with the another chemical that is called amylin, which is secreted along with insulin. And this chemical form amyloid deposits in the interstitial fluid, which also results in the insulin resistance. Materials and methods. The study population consists of 100 human, out of which 50 were healthy persons and they were referred to as control group. Fifty were experimental groups. They were diabetic patients. Anthropometric measurements, weight, height that was measured, and BMI was calculated using the formula. Age, family history, smoking, and exercise habits were evaluated by standard questionnaires. Blood samples were collected after an overnight fasting of 8 hours and plasma was separated. Biochemical analysis. Total protein, albumin, globulin, and lipid profile, liver enzymes, and renal parameters were evaluated from plasma by using biochemical analysis. HOMA-IR calculation. HOMA-IR was calculated by using the formula. Statistical analysis was performed using software SPSS. The data was expressed as mean plus minus standard error of mean and illustrated in tabulated and graphical forms. Now results and discussion. First of all, we will discuss about the lipid profile. The levels of total cholesterol, LDL, and triglycerides were high in type 2 diabetic patients as compared to control, while the level of HDL was low. The reason for this abnormality could be explained in a way that insulin resistance causes the dyslipidemia due to gluconeogenesis, and it will increase the free fatty acids in the plasma. Now, these free fatty acids will move towards the liver and intestine through the blood circulation. Intestine can form triglycerides from these free fatty acids and liver from all type of cholesterols of our body. So there are two types of lipoproteins in our body which carry all type of cholesterol in our body. And these are high density lipoproteins and low density lipoproteins. And in these conditions, when there is a high concentration of free fatty acids in the plasma, the HDL level will be decreased because liver will preferably manufacture more LDL for transport of free fatty acids rather than fluctuating the fatty acids out of the body. So liver will prefer to form more LDL rather than HDL. So all these changes will increase the amount of total cholesterol, LDL, and triglycerides, while the level of HDL will be decreased. The next parameter is liver parameters. In the current study, the levels of AST, ALT, ALP, and bilirubin were elevated than the normal range. AST, ALT, and ALP are liver enzymes, and their increased level in plasma is the main biological marker of hepatocyte damage. Insulin resistance, free fatty acids, and triglycerides accumulation, these are the main contributors of plasma membrane damage of the hepatocytes, which results in the release of these enzymes into the blood. These enzymes are also involved in the process of gluconeogenesis, and due to increased insulin resistance, there is an upsurge in the process of gluconeogenesis, for a long time, which also damages the hepatocytes. Moreover, the gene responsible to produce these enzymes is related to response of insulin in the target tissues. In less responsive cells, the transcription of these genes become elevated. If we talk about the bilirubin, it is the end product of hemoglobin breakdown. But in the liver, there is a protein which is called bilirubin binding protein. It combines with the bilirubin and converts it into unconjugated bilirubin. This unconjugated bilirubin needs glucuronic acid for its release from the body, but due to hepatocyte damage, the liver enzymes or liver cells cannot produce enough glucuronic acid, so bilirubin binding protein will also be diminished. So this will not release bilirubin, and bilirubin will accumulate in the hepatocytes in the liver, and then it will move into the circulation and the rest of the body. That's why the level of bilirubin was high in type 2 diabetic patients as compared to controls. In conclusion, this research proved the association of liver enzymes and lipid profile dysfunction in type 2 diabetes patients. In the present study, dyslipidemia was observed because of long-term presence of disease. High ALT results in the increased sugar level by accelerating the hepatic glucose production, which ultimately adds the complications of type 2 diabetes mellitus. It is well recognized that the surplus free fatty acids present in the insulin resistant condition are directly harmful to hepatocytes. Recommendations. A comprehensive study on the association of liver enzyme and lipid profile with type 2 diabetes in the primary phase of ailment is required to further clarify the picture. Patients on medications and without medications should differentiate to recognize the exact role of liver enzymes. Role of genetic factor, ethnicity, and environment, including diet and lifestyle habits, should also entertain. Genes responsible for the expression of liver enzymes should be studied to evaluate the abnormalities. In the last, these are the references. Thank you very much.